Hi all, my topic is structure of DNA and I am starting with the topic levels of DNA structure. First one is primary structure. It's the formation of nucleotides and their condensation polymerization to form polynucleotide chains. Second one is secondary structure. That is the molecular organization of DNA through the interchain hydrogen bonding and the helical coiling of the polynucleotide chain to form a DNA double helix or duplex DNA. And the last one is tertiary structure. That is the tertiary structure of DNA. It is the coiling and compactation of duplex DNA or DNA double helix to form nucleosomes and then its supercoiling and further compactation to form a solenoid structure characteristic of eukaryotic chromatin. And next topic is Watson and Crick model. So it is explained by James Watson and Francis Crick in 1953. They were honored with Nobel Prize in 1962. They proposed the double-stranded or double helical hypothesis, and it was based on X-ray diffraction studies of Rosalind Franklin and Maurice Wilkins and the Chargaff rules of base pairing. Watson and Crick suggested that the three-dimensional double helical model of DNA structure called bDNA. So bDNA was their model. They revealed that They revealed the fact that the biological functions of macromolecules would certainly be related to their chemical organization. So, in a DNA, there are four base pairs or nitrogen bases. It is adenine, thymine, guanine, and cytosine. The exact molecular organization of DNA is proposed by this James Watson and Francis Crick in 1953. According to Watson and Crick hypothesis, the DNA molecule is in the form of a spiral ladder or a winding staircase. It is formed of two complementary antiparallel polynucleotide chains twisted around a common axis in the form of a right-handed double helix. The two chains are held together and the double helical structure is stabilized by interchain hydrogen bonds between complementary bases and also by the stacking interaction between adjacent base pairs. Each chain is linear and unbranched and is formed by C3 and C5 phosphodiester linkages between a series of nucleotides. The two chains are complementary in that each of them is an exact replica or negative copy of, of the other with regard to the base sequence. They are antiparallel in that they run in opposite directions and have opposite chemical polarities. They have opposite chemical polarities. The terminal phosphate group of the two chains or the C5 terminals of the sugars are placed at opposite ends in the two chains. Moreover, the interchain phosphodiester bonds run from C3 to C5 in one strand and from C5 to C3 in the other. So one strand runs from the 5' end to the 3' end and the other one from 3' end to the 5' end. So in each polynucleotide chain, the constituent molecules are arranged in two series, outer and inner. Sugar and negatively charged phosphate groups are arranged alternately along the other side. They link together through phosphodiester bonds and form hydrophilic and negatively charged backbone of the chain. Nitrogenous bases are arranged along the inner side perpendicular to the long axis of the chain. They stack one above the other in the center of the double helix. Externally, each base pairs or each base links with a sugar molecule through glycosidic bond and forms a nucleoside. Internally, it non-covalently links with a corresponding base of the combining strand through hydrogen bonds and form a base pair. These hydrogen bonds serve as interchain bonds to hold the two strands together and also to stabilize the double helix. The major force that stabilizes the DNA double helix comes from the non-specific and hydrophilic stack interactions between adjacent base pair. So this is the original model proposed by Watson and Crick. The base pairing is peculiar in that it is always complementary. So pairing occurs always between a purine and corresponding pyrimidine, and never between two purines or two pyrimidines. The main reason might be that a DNA molecule should always be 20 Armstrong wide. Pairing between two dicyclic purines would exceed the 20 Armstrong width, and pairing between two monocyclic pyrimids would be short of the 20 Armstrong width. In complementary base pairing, the companion bases are always specific. Thus, pairing occurs always between adenine and thymine, and also between guanine and cytosine. So it is the pairing cytosine with guanine and adenine with thymine. Adenine thymine pairing occurs through two hydrogen bonds, and guanine cytosine pairing occurs through three hydrogen bonds. On account of the specificity in base pairing, the base sequence in one strand will be exactly complementary to that of the other. This makes the two strands complementary to each other. In a DNA molecule, there are four kinds of base pairs. A, that is AT adenine thymine, thymine adenine, guanosine cytosine, cytosine guanine cytosine, and cytosine guanine. These follow no regularity in their number and order. Any pair can follow another. The number and order or sequence of the base pairs represent a precious genetic makeup, which provides the biological information required for protein synthesis, and also for metabolic regulation, and also for gene expression, etc. Thus, the four bases of DNA serve as the four alphabets of the genetic language. DNA double helix has two helical grooves, that is major and minor. The major groove is deep and wide, located in between successive turns of the molecule. Minor groove is small and narrow, and it is seen in between the two strands of double helix. These grooves are believed to facilitate sequence-specific protein DNA interaction. Typically, a DNA molecule is 20 Armstrong in diameter. The average distance between its two adjacent base pairs is 3.4 Armstrong, and the length of a complete turn, that is the pitch, is nearly 34 Armstrong, that is 10 nucleotide residues is located in through a complete turn. So it is all about the structure of DNA, and the levels of DNA structure, and we also go through the Watson and Crick model proposed in 1953. Thank you.